Hello dear viewers, welcome to Mr.Zoologist YouTube channel. Today we are going to discuss about molecular machinery of cell cycle clock. So cell cycle refers to the series of events that occur in a cell's life cycle, leading to its growth, replication and division into two daughter cells. All normal cell types in the body require external signals conveyed by mitogenic growth factors before they will undertake to grow and divide. Since these cells participate in the formation of precisely structured tissues, their proliferation must be coordinated with neighboring cells in those tissues. Cells consult their extracellular environment and its growth regulating signals during the onset of G1 phase until an hour or two before the G1 to S transition. So, if we talk about the restriction point, cells respond to extracellular mitogens and inhibitory factors such as TGF beta only in a discrete window of time that begins at the onset of G1 and ends just before the end of G1. The end of this time window is often called the restriction point or point. It is a point in time when the cell must take the commitment to advance through the remainder of the cell cycle through M phase to remain in G1 or to retreat from the active cell cycle into GO. So that is the R point starting after the M and at the late G1 period during which cell are responsive to mitogenic growth factors and to transforming growth factor TGF beta. However, once these cells had moved through this G1 decision making period and advanced into the final hours of G1, the remaining 10 to 20% of G1, the removal of the external signals, serum, would no longer affect their progress and they would proceed through the remainder of G1 cell cycle and onward through the S, G2 and M phases. Similarly, anti-mitogenic factors such as TGF beta are able to impose their growth inhibitory effects only during this period in the early and the mid G1 phase. Once a cell has entered into late G1, it seems to be useless to the presence of this negative factor in its surroundings. This decision will be respected even if growth factors are no longer present in the extracellular space during these remaining phases of the cell cycle. We know that this series of later steps S, G2 and M phases proceeds on a fixed schedule because a cell that enters S will, in the absence of a major disaster, complete S and having done so enter into G2 and then advance into M phase. Students of cancer therefore focus largely on the GO to G1 transition and the time window when a cell is given the chance or license to make decision about its fate. So, molecular machinery of cell cycle clock is kinases, which are cyclin-dependent kinases CDKs, cyclins, which are the activators of CDKs. The CDKs are phosphorylating enzymes and never act on their own. The CDKs are serine threonine kinases. They depend on associated regulatory subunits, the cyclin proteins, for proper functioning. So biomolecular complexes of CDKs and their cyclin partners are responsible for sending out the signals from the cell cycle clock to dozens of responder molecules that carry out the actual work of moving the cell through its growth and division cycle. The cyclins associated with CDKs activate the catalytic activity of their CDK partners. The cyclins constitute a distinct family of the cellular proteins that share in common a long domain that is involved in the binding and functional activation of CDKs. During much of the G1 phase of the cell cycle, two similarly acting CDKs, CDK4 and 6, are guided by and depend upon their association with a group of related cyclins D1, D2 and D3 that collectively are called the D-type cyclins. After the R point in the late G1, the E-type cyclins E1 and E2 associate with CDK2 to enable the phosphorylation of appropriate substrates required for entry into S phase of the cell cycle. As cells enter into S phase, the A-type cyclins A1 and A2 are replaced by E cyclins as the partners of the CDK2 and thereby enable S phase to progress. Later in S phase, the A-type cyclins switch partners leaving the CDK and associated with another CDK called either CDC2 or CDK1. As the cell moves farther into G2 phase, the A-type cyclins are replaced as CDC2 partners by the B-type cyclins B1 and B2. Finally, at the onset of M phase, the complexes of CDC2 with the B-type cyclins trigger many of the events of the prophase, metaphase, anaphase, and telophase that together constitute the complex program of mitosis. The activities of the various cyclin CDK complexes must be modified or modulated in order to impose control on specific steps in the cell cycle. This regulation depends upon changing the levels and availability of cyclins during the various phases of the cell cycle. So these are the association of the CDKs with respective cyclins. In G1, D CDK4 6 complexes, and after R point CDK2 with E cyclins, and after entry into the S phase, E type cyclins are replaced by A type cyclins, and after entry or late S phase, CDK2 is replaced by CDC2, and in the M phase, A cyclin is replaced by B type cyclins in the M phase, prophase, anaphase, and telophase. So if we talk about the regulation of cell cycle by cyclins, the cyclins for their part also constitute a distinct family of cellular proteins that share in common approximately 100 amino acid residue long domain that is involved in the binding and functional activation of CDKs. The binding of cyclins to CDKs, the association of the two proteins increases the enzymatic activity of CDK to 4 lakh folds. Cyclin A CDK2 complex, such as at the same time the cyclins serve as guide dogs for the CDKs by helping the cyclin CDK complexes recognize appropriate proteins substrates in the cell. So that is a diagram that is showing the activation loop, P state alpha helix, CDK2 and cyclin A, that is forming a cyclin CDK complex. So that is the levels of cyclins regulating the cell cycle phases. Cell cycle B levels are increasing substantially before the onset of M phase, allowing cyclin B molecules to make active B CDC2 complexes that initiate entrance into M phase and that will diminish after the M phase. So that is the level of cyclins in different phases, like cyclin B are active in which phase? They are active in the M phase. In G1, D type cyclins in at the R point, E type cyclins in the S phase is the A type cyclins, and then onset after A replaced by the E type cyclins again. So cyclin E levels are low throughout the most of the G1 phase and abruptly after a cell has progressed through the R point and collapse as the cell enters into S phase. Cyclin A increases as the cell enters into the S phase. Level D type cyclin is strictly lowered and increasing the A type cyclins. For most of these cyclins fluctuations are tightly coordinated with the cell cycle advance through the various cell cycle phases. So collapse of the various cyclin species as advance from one cell cycle phase to the next is due to the rapid degradation, thus being triggered by the activation of ubiquitin ligases which attach polyubiquitin chains to these cyclins. The cyclin's gradual accumulation followed by the rapid destruction has an important functional consequence for the cell cycle because it commands that the cell cycle clock can move in only one direction. So that was the lecture for today. We will meet you in the next video. Till then, goodbye.